Hello, everyone. Welcome to the latest in our monthly seminars that we have here in the Foresight Molecular Machines group. So for those of you who don't know me, I'm James Cooper. So I'm the chair of this group. And for those watching on YouTube later on also, welcome. So this evening we have one speaker, which is Professor Dirk Trauner from NYU, who's going to discuss controlling the machinery of life with photopharmacology. So Dirk, the floor is yours.
Well, thank you so much, James, for your kind introduction, and thanks to the Foresight team for inviting me, and thanks to all of you for attending at what I would consider past my bedtime for some of you. So stay with me. I hope I'll keep you awake with my take on molecular machinery, because I have a sort of a fascination, as all of you, I assume, with the molecular machinery of life. And my take is that instead of building molecular machinery from scratch, which of course has made incredible advances in recent years and decades, I would rather hijack the evolved over billions of years molecular machinery of life and endow it with new functional properties, put on new ignition elements, control elements, switches, to be able to turn it on and off and shape its performance. And we have done this mostly with light. I should say that we're also interested in using magnetic fields, for instance, to do this, and ultrasound, but this is not the topic of today's lecture, of today's talk rather. And please interrupt me at any point to sort of throw me a little bit off track. What I want to tell you about today is the use of synthetic photoswitches and their merger with some of the best studies and best understood molecular machines in cells in order to control cellular functions. But of course, we're also interested in the individual molecules themselves. What I like about this approach is that it's relatively easy to address these machines, right? Because they're kind of wired up through expression in a membrane, and then you can interrogate them, for instance, with electrophysiology and other methods. Let me see whether I can advance my slides here. Yes. So here is a sample of the machinery we have been working on. We started our work some 18 years ago now, actually, with voltage-gated ion channels, which of course make these wonderful movements across the membrane in response to changes in voltage, opening a gate, letting ions flow. And then we have progressed from voltage-gated ion channels over the years to ligand-gated ion channels, such as ionotropic glutamate receptors, such as pentameric ligand-gated ion channels, for instance, GABA-A receptors, nicotinic acetylcholine receptors. Today, I will tell you quite a bit about G-protein-coupled receptors because they have been a wonderful platform for photopharmacology where we put synthetic photoswitches on. I won't tell you much about arguably the best studied, most famous at least, molecular machine, ATP synthase. Some years ago, we published a small molecule, a photoswitchable molecule that functioned like a photoswitchable wrench and was able to control the F1, FO ATPase with light in that way. But we've only done biochemical studies, and the sort of mitochondrial targeting versions is still not ready for publication. So I want to wait a little bit before I can show you some cellular work. But we have also worked on, for instance, on excited amino acid transporters, and we have actually a very nice paper in development with Paul Nissen on CERCA, on the sarcoendoplasmatic reticulum calcium ATPase, which we can also switch with the photoswitchable version of tapsigarginal control. And then we moved over the years a little bit more intracellularly. I have really fallen in love with the cytoskeleton, with its highly dynamic nature and its fascinating movements it allows and enables. And this dynamic instability and dynamic of actin and tubulin, for instance, we could control with photoswitchable molecules. And we'll throw a little bit in today about an attempt to control a kinesin, a spindle kinesin called EG5 with a photoswitchable inhibitor, which is not published yet, but which we are soon going to publish. That allows us to control mitosis with light. And then we did some work on transcription factors, nuclear hormone receptors. A lot of work recently has been done on photoswitchable lipids because these Azobenzene photoswitches, you guessed it, which I'm going to focus on today, they are absolutely a match in heaven with fatty acids. They are lipophilic molecules that can have cis double bonds if you treat them with light. And that's what you find in lipids for reasons. And since we can impart this with light, we can do some pretty cool lipid physiology but also some very nice membrane biophysics with photoswitchable lipids. And there's a whole series of papers in the already published. I can tell you that we have been able now to feed photoswitchable fatty acids to certain cells, and we find that up to 40% of the endoplasmatic reticulum of the phosphatidyl molecules of the endoplasmatic reticulum contain these photoswitchable fatty acids, and therefore we can control, for instance, exocytosis with light in the forthcoming work. But today, as I said, really want to focus mostly on ion channels and GPCRs and throw a little bit in on kinesin. And then maybe, if I get around, also talk about E3 ligases. These are the proteins that mark other proteins for destruction, and we can turn them on or off with light as well. So yeah, the little photo switch I think towards this audience, I don't have to emphasize what is so wonderful about azobenzenes. From my perspective, the fact that they switch so fast that there is no time to fall into triplet states, and therefore there is no singlet oxygen production in biological systems, which actually distinguishes them from other wonderful switches, which are not as useful in biology because you do fall into triplet states occasionally. But an azobenzene just flips within 2 picoseconds, and therefore there is no time for that. And as you know, we can greatly vary them. Some 12 years ago already, we made heterocyclic azobenzenes. So you can change the photophysical properties, the bistability, the thermal bistability greatly. You can change the photostationary states by trying to pull the absorption spectra of the cis and the transform apart as much as you can. Of course, many others named down here have made amazing contributions to that. Also, in terms of sign inversion, Rainer Herges's dazozenes for us were a boon as well. These are the azobenzene derivatives. They are more stable thermodynamically in the dark, in the bent form, and then you can push them towards this elongated form as opposed to a regular or somewhat regular azobenzene. And that, of course, allows us to pharmacologically sign invert in many cases. And I have a whole series of papers in the already published, but also in the making, where this was really crucial. That it actually is a beautiful way to sign invert the logic of an azobenzene, and it works. There are some limitations in terms of action spectra, but other than that, they're wonderful photo switches. And we have contributed to the synthesis as well to make them as synthetically accessible. Yeah, let's classify photopharmacology. The simplest form of photopharmacology is some ligand, for instance, a blocker shown here, an ion channel blocker, that changes its efficacy with light, which, for instance, in the dark is a good blocker. It crams itself into the pore, whereas upon photo switching, it becomes this tau pentagon. This is my cartoony language now. Acute star means active, tau pentagon means inactive, and then ions flow. And that would be a photoswitchable blocker of an ion channel, for instance. And we have made them, and they have actually gone very far. They are now evaluated in a company for vision restoration. But much earlier than that, or somewhat earlier than that, we actually initially tethered the blocker to the channel of interest. And that has one big advantage, namely that the tethering requires some form of bioconjugation, and this bioconjugation can put under genetic control. The simplest form, you can put the cysteine in there, and you do some cysteine chemistry, but as you will see, there are much cleaner and more exciting ways to covalently attach something to a protein of interest or nearby. And in this very early version from 2004, we tethered an extracellular blocker, a tetraethylammonium ion, to the surface of a voltage-gated potassium channel, and with that we could unblock it if the azobenzene was in the trans form, or block it when the azobenzene was in the cis form. This is shown here more structurally. This was this very simple molecule, Malazar, really not so nice nomenclature. I've become better since with acronyms. Maleimide, quaternary ammonium, and azo in between. And that could be tethered to an engineered cysteine on this turret loop of a voltage-gated potassium channel. And then you could either reach the pore or you could withdraw it. This was done together with Rich Kramer in Berkeley. And I have to emphasize that this is the second paper in the history of optogenetics. So, of course, then the channel rhodopsins came along and they kind of wiped us out. But conceptually, at least historically, this is a very early version of optogenetics, and we could make hippocampal neurons fire, excitable cells fire in a light-dependent fashion by controlling this voltage-gated machine with an extra light gate. So a little bit of history here. It's long ago. And then over the years, we have refined this idea of tethered photopharmacology. So as opposed to simple photopharmacology, there's the tethered one. The tethered one requires some covalent attachment, and that typically, not always, because you can also do affinity labeling, but typically that requires some form of modification here, which you can put under genetic control. So, and of course, biologists love that word, genetic control, in addition to light control. So here's the general scheme, cartoon scheme, and here are some molecules. Here, for instance, is a glutamate derivative, with which we control a ligand-gated ion channel, so-called ionotropic glutamate receptor with light. These are truly fascinating molecular machines. This cartoon, while nice, really doesn't do it justice. You have this transmembrane domain. I'm sorry, this is a mistake. This should be TMD, transmembrane domain here. And then you have a ligand binding domain, which looks like a clamshell, like a Venus flytrap. It chews down on the neurotransmitter, on the ligand, on glutamate. And in doing so, this is mechanically coupled with the opening of a gate through which then ions can flow, in this case, sodium in. And that, of course, depolarizes the membrane and leads to firing of an action potential. And that's the major excitatory neurotransmitter glutamate. Right? When you have it postsynaptically released, presynaptically released on the postsynaptic side, it opens these channels through this mechanical event I just described. And we couldn't resist tethering a version of glutamate to the clamshell, to this clamshell domain, as it's called in this world, in such a way that the local concentration changes dramatically from more or less inaccessible to fitting right into the binding site, and then the clamshell closes and the channel can open. And this is also ancient history now, because this required cysteamylamide chemistry, and we refined this chemistry somewhat. For instance, we went from this azobenzene diazyl aniline to this azobenzene, which is a push-pull system, which has a electron donating substituent here and a sort of neutral, more or less neutral substituent here, slightly electron withdrawing perhaps. And then in doing so, you can greatly shift the action spectrum from a lambda max of in the UVA range to one in the blue range. And in fact, when we look at the action spectrum, so it is an occurrence plotted as a function of light or darkness, because this thing also turns itself off automatically, since it's a thermally unstable one. It's a fast relaxing azobenzene. You can see that we have a very nice action spectrum. And I like this because this action spectrum pretty much matches the action spectrum of the blue cone in the retina and in trichromatic vision. It's pretty much, it's almost identical actually. And yes, indeed, these things have been used to restore vision, the systems, but not for very long because something better came along. And this something better I want to illustrate with the metabotropic glutamate receptor. The metabotropic glutamate receptor now is a G protein coupled receptor, but it's dimeric, and it has also this extracellular venous flytrap like ligand binding domain, LBD, which is actually quite similar to the one in ionotropic receptors. This is one of these motifs in nature that has been picked up again and again and again. This sort of chewing down on a ligand has been adapted to a wide variety of receptors, ion channels, GPCRs, also receptor tyrosine kinases, transporters. Some ABC transporters have this motif. It's a typical case of miss and match in evolution, I believe, that leads to giant leaps in functional sophistication, changes at least. So, and it's dimeric, so there's the 7-helix transmembrane domain, and then the ligand binding domains are actually linked together by a disulfide. So it's actually dimeric GPCR. It's a family C GPCR, if you want to classify it. And that chews down on glutamate, and then this leads to intercellular signaling, but not flow of ions, rather dissociation of the heterotrimeric G protein. But ions do flow in the end because you also activate GERK channels, G protein coupled inward rectifier channels. And as such, this is one of the major modulatory metabotropic receptors. And we have initially done some simple photopharmacology, not so interesting. We've done some closely tailored photopharmacology, not so interesting because this systemalamide chemistry works nice on cells and in some simple tissues, perhaps also in the retina. But it is due to the hydrolysis of the maleimide, not great chemistry for bioconjugation, as shown here. So this is the bioconjugation, but it turns out that these maleimides, of course, are not indefinitely stable in serum. But there's a much better way to do this, and this is what I want to mostly focus on. This is bioconjugation through bioconjugation tags. These are essentially suicidal enzymes, proteins that self-label, that are engineered to covalently attach an electrophile with the cleanliness and the speed of an enzymatic reaction. And this electrophile is not otherwise reactive, does not react with water or with glutathione. It's a very poor electrophile, but once it's captured by this enzyme, it will lead to a suicidal enzyme labeling, so to speak. And arguably the most famous ones are the SNAP tag and the CLIP tag and the HALO tag, which react with benzylguanines or alkyl halides respectively and have been greatly engineered and can be fused to all kinds of proteins or nearby, placed nearby proteins to now allow for the covalent attachment of some tethered molecule. And as opposed to the previous closely tethered version, this tether has to be pretty large and flexible because these are not so small. These are sort of 22, 24 kilodaltons, these domains. And therefore, you don't really change the local concentration much with the photoswitch. You move the photoswitch into the pharmacophore, as indicated here, and then you change the efficacy of the pharmacophore, but not so much the length of the tether and the orientation of the tether, because this is a long polyethylene glycol chain. We call this portal for photoswitched orthogonally remotely tethered ligand. So you see we got a little bit better with our acronyms too. And it turns out this portal concept is indeed very broadly applicable. You will see a lot of varieties in a moment. But the important feature really is that the switch is in the tethered pharmacophore. I always compare this with a dog on a leash, where the leash kind of restricts the action radius of the dog and tethers it to an owner of interest, but the bioactivity is in the snout, right? That's where it happens, as opposed to a free diffusible dog. This, by the way, was in May 2020, just to give you an idea what New York looked like in these early days of the pandemic. It was eerily beautiful in a way. And we have been able to apply this portal concept now to a very broad variety of receptors. We've done it with an ion channel now. This is unpublished. We've done it with glutamate receptors. We've done it with dopamine receptors. We've done it with serotonin receptors. You'll see a little bit about this, also unpublished. So family A G-protein coupled receptors and family C G-protein coupled receptors. And here's the metabotropic glutamate receptor version that where we N-terminally extend the ligand binding domain with a SNAP tag or some tag. And then we attach the photoswitch on a leash. And if the photoswitch gets activated with light, it activates first the ligand binding domain, and this is mechanically coupled to a change in the interface here. Leads to dissociation of the heterotrimeric G protein, and this can be a G alpha I, for instance, in the glutamate receptor, metabotropic glutamate receptor inhibitory. The G beta gamma then travels and activates G protein coupled immune rectifier potassium channels, which also have an inhibitory effect. So these are mostly inhibitory receptors. And that actually worked beautifully. A little bit of chemistry. This is the tethered glutamate. Here's the photoswitch. Here's the glutamate with a specific stereochemistry here, or configuration rather here, should be precise. Here we have the long, more or less long flexible tether. We have also longer ones, shorter ones. We put this together. You don't need to have click chemistry. This is just cute, but of course you can just make an amide because you make it in vitro anyway. and then you add it. And here's the benzylguanine. And everything that is on this benzyl group gets transferred to a cysteine in this NAP tag, but only to that cysteine, nowhere else, because this is more or less unreactive, also with glutathione, and is a special electrophile in that way. And we have made one that maximally activates with 380 nanometers with UVA light, which is maybe not so good for in vivo work, but it is very nicely compatible with fluorophores. So there's that. And we also made some that is activated with blue light for 60 nanometers maximally. And the only change, let's go back, look carefully here, was the removal of one carbonyl here. So this one carbonyl here makes all the difference in terms of the action spectrum. And here's a model of what this looks like. Before I rotate it, I just want to explain, these are the 2 7-helix transmembrane domains. In the meantime, by the way, there is a cryo-EM structure of, I think, mGluR5 and a few other mGluRs. And then here's an adapter, a so-called cysteine-rich domain. This is just really a lever. This is a very stiff adapter, mechanical adapter. And here is the clamshell, the 2 clamshells sort of back to back. When I rotate this, maybe let's rotate it, you will see it better. The 2 clamshells, and as the clamshell closes, this lever arm here pulls on the 7-helix transmembrane portion, which then changes the interface and kicks out literally the heterotrimeric G protein that is attached here, which then travels and does downstream signaling. And let's maybe go back here. And in orange, I have modeled in the snap tags. You get the sense how small or large the snap tag is. And in green, we have modeled in the tether and the photoswitch. And here in red, this is actually glutamate, which was from the crystal structure. So you get a good sense for how big this is. The sphere, the 5 nanometer sphere here, we put in to sort of measure or calculate the local concentration, effective concentration of this glutamate, which is very high. We're always in this sort of high millimolar range, mid to high millimolar range. And this construct was expressed by Udi Isakov and by John Flannery with a viral transfection method with an AAV virus in the retina of blind mice. And then Mike Berry, the graduate student with Udi, who is now at OHSU still, I believe, he actually measured with multi-electrode array recordings these electrical activities from a blind retina. So this animal was blind. This is a blind mouse model, and you can explant the retina, sacrifice it, explant the retina, and then measure extracellular field potentials. These are these little sort of ticks here, which you can sum up, and you get a sense for the electrical activity of the retina at the level of retinal ganglion cells, which are the sort of output neurons that project the nervus opticus. And you see when we express this LIGLUR and this portal version and add our molecule, we see very nice inhibition when we irradiate with 460 nanometers. So there's clear inhibition. And this happens within 1 second, but it also happens at much shorter time scales, 500 millisecond. Oops, let me maybe try to make a nice laser pointer. For some reason, I always lose this. 250 milliseconds, and all the way down to 25 milliseconds. You see a very clean signal. And I like this because that's kind of the time unit of psychological processing, of visual processing in humans. If something happens at a rate of 50 hertz, 20 milliseconds, we see this as a continuous picture. The older colleagues here will remember self-refreshing screens, right, which were refreshing at a rate of 20 hertz, and you could still see a movie on them. And I don't know what the frame rate now is in a movie theater, but in the old days it was about that as well. So that's kind of the unit, and I find this very cool because in 25 milliseconds, this means that the photoswitch switches. That's a very fast process. I already said that. It's about 2 picoseconds. And then the now activated ligand finds the clamshell, or the, let's call it Venus flytrap. The flytrap closes. This leads to dissociation, travels all the way down here. The signal leads to dissociation of a heterotrimeric G protein. All this stuff happens that leads to inhibition. Then as soon as the light is gone, this kind of spring-loaded system pops back into an inactive form and turns itself off. And all that in 25 millisecond which of course biochemically is an eternity, but I still find this cool from a machinery point of view, so to speak, that this is so fast. But it reflects really the high local concentration, the likelihood that this in the activated form immediately finds its binding site, and then as soon as it's relaxing thermally, it dissociates very fast. So the decay off must be very fast as well once this gets into the inactive form, relaxes in the inactive form. Maybe we got just lucky, but it turns out to work quite well. Okay, let me take a quick sip from my coffee because I'm talking fast. So how can we increase the effect of this? Well, my student Johannes Bochhagen, who pioneered this work in my group, they moved on and refined it and came up with this very nice idea to make branched tethered portals. I like to call them the Kraken, right, or the Hydra, because that further increases the local concentration. Maybe let me go to that molecule right away. This is one that Johannes made with 4 of these photoswitchable molecules now on, and I always like to compare this to the Hydra. It's almost impossible to beat this down anymore when you are a receptor, when they're going after you, and that's what you see here, right? Because now the response upon irradiation is as strong as saturating glutamate. This is 1 millimolar saturating glutamate. And you see that with this, we can now do it, and you can multiplex it. You can have different photoswitches with different light sensitivity. This is all in development. We can add fluorophores to also see where this stuff goes. We can add antennas and so on, because this bioconjugation chemistry is very robust, and these are still, compared to proteins at least, or nanobodies or something like that, relatively small molecules that easily diffuse through the system. The other thing that we thought about is can we do this with native receptors? So one problem you have when you do genetic manipulation and is that you overexpress something unnatural, often towards the background of the natural receptor, and that complicates the analysis. And controlling these expression levels is not so easy, and therefore it would be great if you could actually control native receptors with light. Going away now from genetic control because we are thinking more about therapy now. And of course, simple photopharmacology works, right? This is clear that you can do this. But if you want to do it a little bit more sophisticated, you could think about nanobody photoswitch conjugates, NPCs, which we already have pioneered and more in development, because then you have a very nice non-covalent but strong interaction here. You can also place the portal bioconjugation tag onto a protein nearby. We had a paper with Udi Isakov last year where we call this MA portal, the membrane anchored portal. And this actually works beautifully because the lateral diffusion of these systems is fast, and the local concentration is still high enough, and you still have the possibility to anchor the pharmacology to a cell that you have defined. So this is a great way to do selective pharmacology with native receptor expression levels. Here's a version of this nanobody one that I have published already. We teamed up with Johannes Bochhagen and Josh Levitz here in New York. And Josh has made this GFP fused metabotropic glutamate receptor. And there was a nanobody SNAP-tag conjugate already published. So we just had to make this, make a nanobody. Published was the crystal structure of GFP with this nanobody. It was a good nanobody for GFP, and we just N-terminal or C-terminal extended it to make to put a SNAP tag on. And although this looks unwieldy and complicated, this worked actually beautifully. We could control metabotropic glutamate receptors, in this case, slightly modified or GFP tagged with this nanobody. Should update this. This has long been published. All right. Let me now dive a little bit deeper into sort of machinery considerations and what I like to call synthetic biology. I already told you what I love about nature's molecular machines is that they are modular and you can mix and match. Right? This is very often the case that a certain motif that has been functionally used can be combined in different ways to confer new functions. And we decided to do this too we decided to turn the human insulin receptor into a photoreceptor, but first into a glutamate receptor. The human insulin receptor is arguably the best studied receptor tyrosine kinase. This binds to its alpha-beta subunits here to insulin, and then this leads to cross-phosphorylation, kinase activity, and then you have downstream signaling, usually through the MAP kinase pathway. So classic, of course, in signal transduction, widely studied how this exactly works. But it's dependent on a conformational change on the extracellular side upon insulin binding. And Johannes Bohagen and Philipp Leipe in my group in Munich still decided to just cut this whole thing out and replace it with the venous flytrap domain of a metabotropic glutamate receptor and the cysteine-rich domain, by the way. I'm not showing it here, but this lever, this stiff lever was also there. It was actually very important. And they make a fusion protein that now has a glutamate binding site as opposed to an insulin binding site. And amazingly, this actually could be turned on with glutamate. Not very well. We used close to 100 micromolar, 1 millimolar glutamate, so high concentrations. But we could actually turn on the insulin receptor. And of course, the downstream pathways of the insulin receptor are very well studied. You can, all kinds of antibodies against the phosphorylated proteins, including the receptor itself, available. And therefore, we could study it. And once we had established that, that was actually the hard part. I think Johannes made about 12 versions, and 2 of them worked, 12 constructs, and 2 of them worked. We also teamed up with Jérôme Vinon in Lille. And once we had this down, of course, we knew how to turn this into a photoreceptor, because that's, again, mix and match. This is our modular system. So we put the SnapTag on the photoswitch, and lo and behold, this is now a human insulin receptor that can be turned on with light, at least in xenobus oocytes. Don't know yet for sure what it's good for, but as a sort of basic scientist and a biomolecular machine aficionado, I think that this is quite interesting. And here are the data. So sorry for the information richness, but let me point out that only when we have here in the insulin receptor, we call this LI here, the light-gated human insulin receptor, only if this thing is expressed and the molecule is present, we can do a lot of controls. Then we see cross-phosphorylation of the receptor itself, and then downstream phosphorylation of ACT and downstream phosphorylation of ERK here with through the human insulin receptor. And we can also do this with another receptor tyrosine kinase called MET. The same thing, we see cross-phosphorylation, autophosphorylation with immunoprecipitation of the receptor itself, and then phosphorylation of ACT and of ERK with these phosphor antibodies. So this was published together with Jérôme Vicon and Colette Dissou late last year. So an exercise, I would say, in synthetic biology. Now let me wrap this up, at least this part, and then you tell me how much time I've left, by telling you a little bit about family A GPCRs. These are the somewhat simplest one because they don't have these large extracellular domains. The most famous one is the photoreceptor, namely the opsins, rhodopsin, which has a covalently tethered inverse agonist bound to a photoswitchable one. Bound, right? But that's the only photoreceptor, that and the melanopsins. So the opsins are the only photoreceptors in the human genome, as far as we know. But it's pretty clear now that they're the only ones. And we were very interested in imparting the logic of opsins to other GPCRs, such as adrenal receptors, metabotropic receptors, acetylcholine receptors, for instance. And opioid receptors, you name it, lipid receptors. And the simple way is, again, simple photopharmacology, where you make something acute and then it activates the receptor. Or you can tether. And again, the huge advantage of tethering is that you can now untangle pharmacology. And in unpublished work that's going to come out very soon, we have now finally found a good way to do this with the serotonin receptor, 5-hydroxytryptamine receptor, 5-HTR. Serotonin is a very complicated neurotransmitter, neuromodulator, because it has a lot of receptor subtypes. And this is very difficult to untangle. On the other hand, they are hugely important in psychiatry, right? 5-HT2R, for instance, is the one that responds mostly to LSD. It has psychedelic effects. And they are right now in the forefront again for depression and anxiety and all kinds of diseases. But they are really difficult to selectively address with selective pharmacology. However, if you can tether the pharmacology through your receptor, through a subtype of the whatever 14 subtypes there are, then you will be able to gain more insights in what the exact role of this is. And if you can put it in the light control when it's otherwise silent, it's even more powerful. So we have extended the serotonin receptor 5-HT2R. I should have put this down here. 5-HTR2A, the subtype 2A, with a snap tag, and we have tethered the photoswitchable version of serotonin to this receptor. And as you can see here from these calcium traces, we can very nicely turn this receptor on and off. Here it's written out 5-HT2AR. That's the correct way to say it, with the snap tag. And a benzalguanine version of our photoswitchable serotonin. So this is a tethered version now on a leash. And we can very nicely, as seen from these calcium traces, these are GQ coupled, so you can look for calcium traces. We can see that we can irradiate with 380 nanometers to turn this receptor on and off. And here is saturating 1 micromolar 5-HT, 1 micromolar serotonin. So stay tuned for this. This will probably be put online next week. But we are very excited because, again, the untangling of serotonin receptors has been a nightmare and has been very difficult. But tethered photoswitches provide a solution. How much more time do I have?
You've probably got about 10 minutes.
10 minutes. Then let me briefly talk about another unpublished work, which really works on motors, right? So molecular motors I haven't talked too much about. And of course, for this audience, this is unforgivable. And some of the most famous motors are kinesins. And in terms of pharmacology or medical importance, one of the most important kinesin is the mitotic kinesin EG5. This is the one that pushes the centrosomes apart in mitosis. In the early phase of mitosis, the centrosomes need to be pushed apart, and then the chromosomes need to be lined up at the equatorial plate before they can be pulled apart. And this movement is conferred by EG5, by the mitotic kinesin EG5, which is a tetramer, and links to these antiparallel microtubules on both sides, and then literally pushes them apart. And therefore, the mitotic spindle, the bipolar mitotic spindle forms. If EG5 is inhibited or depleted, cells have this phenotype here, this monoaster-like phenotype, monopolar phenotype, where the microtubules sort of project from a central, you could say tetramer or double dimer of two centrosomes, and therefore it's very clear what happens. This is a typical phenotype of EG5 inhibition or depletion. And there are some famous molecules such as monastrol, as in monoaster, that confer this. And since this happens in dividing cells, of course, this has been explored as an anti-cancer strategy. Unfortunately, so far, nothing is on the market, but it has certainly been looked into. And we decided to make photoswitchable inhibitors of the mitotic kinesin EG5 to complement our work on the optical control of tubulin and actin. You may be aware that we've published a couple of papers now on tubulin and actin with photoswitchable versions of Taxol and of compretastatin and of jasplakinolide and so on, of latrunculin. But these are the tracks, right? And here are the motors that work on those. And we finally, after actually a very long systematic search, this was a tough one, we found a molecule that has this property that is a photoswitchable inhibitor of EG5 in the cis form of the azobenzene. This is a classical azobenzene, so 370 nanometers to bend it to the cis form, and you can push it back to the trans form. And we initially always do live dead assays just to see whether this works. These are not super informative, but at least to get a sense for whether there's an effect on the cells. Depends on whether you can push them ultimately into apoptosis or not. And you see very nicely, and this is important for us, that the compound is more active as an inhibitor in the cis form than in the trans form. And then when we do cell sorting, fluorescent cell sorting, we see this really beautifully what happens, because when we add this molecule and irradiate it with 370 nanometers, we push pretty much for almost all of the cells into the G2M phase of the cell cycle. Whereas if the molecule is in the trans form, in the dark, it's a normal distribution that you have with DMSO either in the dark upon irradiation. So we can very nicely push this into cell cycle arrest in the G2M phase. And we can also do imaging. And we see here, for instance, that with a parent molecule called EMD, with a number which doesn't have the photoswitch, you see these monastral phenotypes. So this is clearly an EMD EC5 inhibitor rather. With our ASO version, we see that only with the 70 nanometers, we push them into this monastral phenotype, whereas in the dark, the chromosomes line up nicely and the bipolar spindle is formed. These are the chromosomes here, these white bundles. And DMSO, of course, is just the control. And together with Henry Hess, I wrote this slide half an hour ago, so Henry Hess's name should be here, who of course is very prominent in our community. We showed that in gliding assays, that we can also look at it biochemically. So here is the inactive form, the trans form. Let me actually show this again. The trans form, you see that these are trashing, these are relatively mobile, and we capture many of them. Whereas if we go to the cis form, we capture very few, and those that are captured, that are actually in the plane, are very little active. And there is the statistics, which I'm going to disclose in the paper. I think I'll stop here because I could tell you now about E3 ligases and their fascinating similarity to alien spaceship machines, but that's maybe something for a different day. We can control the degradation of proteins with light through a photoswitchable PROTAC that we call PHOTAC. And just leave it at this teaser level. Thank you.
Well, thanks very much, Dirk, that was like a crash course in biology and synthetic biology, at least for me there, with all the structures and everything that was going on. There's a question in the chat from Thomas Schroeder. What we've done in the past is had you engage with the chat because it seems to be easier than me misunderstanding the question, if that's right. There's only one, and then people can unmute themselves, and I might ask a few as well afterwards.
How do I do this again? How do I, after 1.5 years, I still ask, how do I do this?
Well, I can just read. We'll break with tradition. I can just read it to you. So the question was, am I right in understanding that the advantage of remote tethering over close tethering is a matter of being able to selectively address a molecular machine of interest over others in the system via genetic engineering? Absolutely.
Both closed and remote tethering. I would say the closed tethering, let's forget about it. I'm not doing this anymore. The system alignement chemistry is yesterday's news because the what I call remote tethering actually works so well, right? You either tether it to the machine of interest, so you can, I don't know, at some point fuse something with a subunit of F1FO, right? And then throwing that range in a light-dependent fashion, and you can only do this now with versions of that machine that you have genetically tagged, or you do it nearby, either with a membrane-anchored protein that is close and that has a pretty high local concentration because it has only lateral diffusion. But you can also think about scaffolding proteins, for instance, that you use as your docking station for attaching the tether. We haven't shown this yet, but I think this is something, especially in the synaptic cleft, that one should explore. So, and yes, the real advantage is that we can determine which subtype, or at least which cell type we are addressing.
So I don't know if other people have questions, maybe they're still digesting everything that was in the talk.
The gliding assays again. Do I see this correctly? Is that what you want to see again? Okay. Is that what you want to see again, Ted?
Yes, please. So tell us a little bit more about what's happening in these two movies.
So this is immobilized egg 5, which moves tubulin that is fluorescently labeled. These are these gliding assays that are in the community of motor proteins quite common, that you immobilize a motor protein, and there's some sophistication in how to do this correctly. And it turns out with egg 5, this is not so simple, because egg 5 functions as a tetramer, right? So there's some sophistication. And then you, like in a mosh pit, you literally move your rock star along this immobilized one. But you first have to capture them, and then you can move them. And what we see on the left side is that first of all, we capture a lot of these microtubules, and then they are somewhat mobile. Whereas on the right side, we actually capture very few, and those who are captured, the ones that move so fast are actually not captured, but those who are captured, they are not moving very fast. And I have to admit, I have to defer to Henry Hess for the fully professional explanation. That's my understanding. But the statistics are in the paper. And Henry does this for a living. I know this is a lame answer, but that's the gist of it. You stabilize, you make these microtubules by also adding Taxol to sort of prevent them from depolymerization, and add a fluorophore, and then you can actually visualize those.
Yeah. So one of the main questions this group has for most of our speakers is to sort of speculate a little bit on like 5, 15 and 30-year goals inside. Maybe it could be in the general field, but I suppose in this it would be your sort of specific area of research, perhaps leaning towards, you've discussed a bit about synthetic biology. Do you have some idea of where you think the work in this field is going?
There are medium-term goals. I already talked about the possibility to control things with other input signals than light. Light is great, right? But light doesn't go far through the skull. And if you can do this with magnetic fields or with ultrasound, then perhaps, and I think, is Harald here? I think I saw Harald. I don't know whether it's the Harald. I saw a Harald here. He can answer this a little bit more. Is certainly more farther along with adding additional control elements. I would love to put molecular motors on proteins. I don't see a reason why one cannot pack a Feringer motor onto a snap tag and then move things around in a sensible way, right? And for me, sensible always means that it has some interesting biological function. But if you have I'm speculating here. If you have a membrane anchored snap tag and you make a benzylguanine Feringer motor and you pack it on, I'm pretty sure you can move things along the membrane with light. And that would be interesting, right? I mean, forces have been generated by making gases, right? This is well known. But other than superoxide dismutase, I don't know too much force generation that has been used in biological functions, artificial force generation.
And then one thing that I would ask, so I'm a synthetic supermolecular chemist. I have a background in things that operate in membranes, and that's where my independent career is currently sort of focused. And so I think about problems in this area from a totally synthetic point of view. So my question would be, so your talk today has been focused on integrating synthetic elements with biological constructs. Exactly. Do you think, it's sort of leading on the question, do you think that's the real future of molecular machines, or do you think there's a scenario where we can have a totally synthetic analog of what biology can do?
Absolutely, so it's just not my personal take. I absolutely believe it. Will it be at the time scale of Kitty Hawk hop to Boeing 747. We will see, right?
Yeah, if that happens, I'll be 100 before we finish, which sounds about right. So,
right. So we will see. But you know, it's clear that we have made already enormous strides. Could you design something as complex as F1FO from scratch with current knowledge? I'm not sure. What I really like, it's not so much that I say, oh, nature is 4 billion years in, and how can you compete? On the contrary, I actually don't believe that is all, right? Because the developmental cycles are getting faster and faster, and we are learning more and more. And at some point, we have machine learning in this field, and already have it. And what I really like about these biological machines is that they are so addressable, because you can link them up with something that you can address. So the wires, the big problem, in my opinion, with synthetic molecular machines is where are the wires that you can address them with, right? And how can you immobilize them in a way that they perform work that is useful and can be multiplied and whatnot. And this is all with transmembrane proteins, and that's why we started on transmembrane proteins. This is all a given, right? You can you already know how it is oriented, and you know how to visualize it, and you know how to address it. And I think that is important to have sort of the supramolecular environment. You have to think about the environment where you place your molecular machines, and this is not done enough in the purely synthetic community, if I may say.
I mean, I think from a membrane point of view, it's very rare anyway, just as a general concept. I think that's something that a lot of people in this community are very, including myself, very interested in.
Yeah, you can do COFs and MOFs. You can do things that membranes can't do. You can do COFs and MOFs and things like that to place stuff, right? And that is extremely exciting, I think.
So I don't know if there's any other questions from anyone in the audience. There's certainly none. I don't think there are any others in the chat. So I don't know if anyone wishes to either just unmute themselves or hands up. Otherwise, we can.
I feel I haven't talked enough about the sophistication of the azobenzenes, because we actually think a lot about the molecular details of these azobenzenes. I just alluded to in the beginning, but we are following this very closely. What can be done with azobenzene photoswitches? How can you push them into the near infrared? Right? We have now azobenzene photoswitches that we can integrate in lipids that work with C60 nanometers. I just had a paper on lipid nanoparticles that are made up of photoswitchable lipids that you can release cargo with C60 nanometers. So that's a molecular or a synthetic front I'm very closely following. There will be clickable azobenzenes more and more, right? This is coming online now. Our first paper is out on using click chemistry and using the modularity of click chemistry to make photoswitches. And I think this is going to be exciting. And it's not that I'm not in awe of the other photoswitches, but as I've said, we actually worked with some of them. Spiropyranes, for instance, in biological context, there are issues with bleaching and single oxygen production.
Yeah, so I wonder if that's it. If this is the last chance for anyone to ask anything else, if they want to. Otherwise, we can wrap up here, I think.
Yeah, what I wish I would have more input from is the modeling community, right? The molecular, because I know that this is getting very strong in molecular machines, and what I showed you as a model is what just cobbled together. But if I wish somebody would. We had more contact with the molecular dynamics community to really understand. There are so many questions we don't understand. We don't understand, for instance, whether the photoswitches not well enough, whether the photoswitches can photoisomerize whilst bound to their receptor, or whether they have to come off from the receptor. And I'm not biophysicist enough to systematically look at that.
There's a call there for anyone in the audience that might have that level of expertise. I suppose I think it's something that I certainly would benefit from as well. If that's about it, there's nothing else in the chat that I can see. I don't see any raised hands or anyone else wanting to to ask anything. So we thank you very much again for sharing the work with us, Dirk. It was really exciting and interesting stuff. So we'll call the meeting to a close there. And thanks everyone for coming along. I think it's been really interesting today to see a bit more biologically applied chemistry, which I think we haven't covered quite as much in this, although we've had some in the past. So yeah, it'll be the 3rd week of August next month for the next talk. So thanks everyone again, and take care. So bye-bye.